Good afternoon. Welcome to this webcast. I'm your host, Jennifer Wicks from Learning and Development. Very happy, as always, to work with the research team in STEMnet here at the Office of the Chancellor. And today, of course, it's NSF Career Awardees. The presentation today, we have many presenters and experts from across the CSU system campuses ready to share their body of work with you. So we look forward to your comments and questions over in the open chat pod, which will be on the left side of the screen. And we'd like to know a little bit more about you before we get started. Want to know, what are you most interested in learning in this session today? And also, which campus are you representing today? Which campus are you joining from or affiliated with, as we're all working from home? We'd like to know where people are joining from across the system. As you can see, we will have live captions throughout the session as well, and look forward to an engaging Q&A segment at the very end of the session today. Today the session is a full 2 hours, so we'll be going presentation to presentation from 2 o'clock now over until about 3:30 or 3:45, and then we'll have a really nice discussion with the Q&A facilitated by Dr. Frank A. Gomez. With that, I'd like to introduce our facilitator, our moderator today, and Dr. Frank A. Gomez, who is the Executive Director of STEMnet at the Office of the Chancellor. And with that, I will hand it over to you to begin, Frank.
Jennifer, thank you for the kind introduction, and welcome to all of you to another STEMnet webcast. I hope you and all your families are all healthy and safe. Today's webcast is another exciting one, focusing on several CSU awardees of the prestigious National Science Foundation NSF Faculty Early Career Development Career Program. The Career Program is a foundation-wide activity that offers the NSF's most prestigious awards in support of early career faculty who have the potential to serve as academic role models and who exemplify the role of teacher-scholars through outstanding research, excellent education, and the integration of education and research within the context of the mission of their departments and institutions. Career awards provide recipients the opportunity to enhance their professional career development, better integrate their research and education responsibilities, and build academic leadership abilities. In 1997, I received a career award and understand the role it can play in a faculty's career. At the time, I was unaware of the level of importance in receiving an award, and so too neither did my institution. Career awards to the CSU were few at the time, perhaps only a handful a decade. Today, the number of faculty receiving across the CSU garnering the awards numbers roughly 3 to 5 per year. This increase demonstrates the high caliber of the CSU faculty, their ability to compete with R1 institutions, and an NSF and reviewers' recognition of our world-class faculty. CSU faculty are prime examples of the teacher-scholar model, and COVID-19 or not, it is important that the CSU do its best to support outstanding faculty, like all of you, in their support of outstanding research and education and their integration across all 23 campuses. Today it gives me great pleasure to bring together a sampling of our past and present CSU NSF Career Awardees to speak on their work and to provide a glimpse of what it has been to be a career awardee. The career program opens up opportunities not only for faculty, but for our students, and provides them with a great experiential research experience. Before we begin, I want to remind the audience to use the open chat box on their screens to type in their questions. Our speakers, when not presenting, will do their very best to respond in real time. So let's begin. I look forward to hearing from all of our presenters today. Our first speaker is Chantal Stieber from Cal Poly Pomona. Chantal, the floor is yours.
Hello everyone, welcome. Today I'm going to talk about building a research program for targeting pollutant reduction from small internal grants to an NSF Career Award and a brighter future. So I won't talk too much about the project, but for those of you who are chemists and are interested in learning more about it, I'm happy to chat at a later date. I unfortunately won't be able to make the Q&A today, so feel free to email me if you have questions about anything. So for the career project, my project had 5 aims. The first 3 aims related to my research, and aims 4 and 5 related to teaching more specifically. So aim 1 was developing X-ray emission spectroscopy to characterize metal NOx species. So the NO species are common pollutants. Aim 2 was to advance knowledge of X-ray emission spectroscopy through computational methods. Aim 3 was to synthesize new metal complexes bound to NOx species and establish spectral trends. So here you see in my lab, we have 3 components. We have synthetic work, spectroscopy, and computational work. The 4th aim was to introduce computational inorganic chemistry research into the classroom, and the 5th aim was to implement hands-on crystallography in education. With regards to actually setting up the career proposal, I wanted to give you some more specific information on some tips. So I really leveraged the wonderful internal grant funding opportunities before applying for the career grant. So I was able to get the RSCA grant, SPICE, Teacher Scholar, Exceptional Service, and my students have been very successful in getting various research fellowships, so through McNair or RISE, LSAMP. In my second year, I was able to get CSU level funding through CSUPERB, so $15,000. This was really critical because it actually allowed me to get more funding to be able to do off-site experiments. We do experiments at the Stanford Synchrotron Radiation Light Source, and through that, we were able to get a paper published before the career grant and get a lot of preliminary data. We also utilize NSF Exceed computational resources. These are quite easy startup grants to get, so if anyone's interested, please reach out to me. I'm happy to connect you on how to use these. And I applied to NSF twice before I received my career. So the first I applied was NSF RUI, which is the funding mechanism for PUI institutions. I applied for career once before I got it. And then I applied for the American Chemical Society Petroleum Research Fund 2 times and was unsuccessful. The big grant that was probably instrumental in getting the career also was a Department of Defense instrumentation grant for single crystal X-ray diffractometer. So this was $600,000. This instrument is used to determine 3D atomic structure. And so it's really critical for moving our research forward. This is also a funding mechanism that's specifically for historically Black and minority serving institutions. So again, something that the CSU really can leverage is getting some of these instrumentation grants. So I just finished my first year of the career grant. In terms of deliverables, what we were able to achieve, we trained 15 undergrads and 3 master's students. We had 2 publications published during the time, one in the Journal of the American Chemical Society, which is the top chemistry journal in the world, a crystallography paper. So here on the right, you can see a 3D atomic structure of a molecule we were able to make. A 3rd publication was submitted and has since been accepted. For teaching, we created a new course in crystallography, made 3 training videos. This was all before COVID, so it's actually been very useful to have the training videos now. We did data collection at the Stanford Synchrotron Radiation Light Source, and my students presented 17 presentations at 3 different conferences. So the other thing I wanted to share with you is that I actually had the opportunity of serving on a career panel, and so I've been able to see kind of both sides of the spectrum. The first thing I'd say is that most proposals are absolutely excellent. The panelists are really looking at small differences, really nitpicky things, and in the end it comes down to feasibility and readability. So you want to have a proposal that's beautiful and pretty and aesthetically pleasing so that it's easy to read and view. And the ones that were really beautifully written and visually pleasing in your first pass through already seem to be better. Figures help a lot. So here on the right I actually have an example of a figure that I modified for my second time around. So one of the challenges we run into at PUIs is we often get dinged. Either our research is too easy and so not interesting enough, or it's too hard to do at a PUI. And the latter is one that you can really target through good figure making. So here on the right, in my original proposal, I only showed the mechanism, so the two complexes on the right. For the second year, I actually added on the left that structure, which is 3D structural data, and I specifically wrote X-ray structure by third year undergraduate to really hammer it into those reviewers that we can do this, undergrads can do this, and we can do this at an undergrad institution. You want to reiterate important components, so if there's something that is really critical to your research, put it in the proposal multiple times. The reviewers didn't write the proposal. We might miss it if it's only in there once. But if it's in there in every section, we won't miss it. You want to be specific, so really have a detailed plan on what you're going to do. And the other component in terms of feasibility is that preliminary results are absolutely key. So the people who really were competitive were ones who had a paper. I wouldn't recommend applying before you have a paper, or at least something submitted, maybe on an archive like ChemArchive or BioArchive. And you really have to show that you're able to do these experiments and talk about what type of data you've already been able to collect. You want to have a well thought out teaching plan and broader impacts. Here, I think for PUIs, we tend not to have as hard of a time with it. But again, it's important to have concrete evidence. So really, if it relies on a collaboration, make sure you have those collaborations established, that you've talked to those people, you have a clear plan on what you're going to be doing. In my case, in my first round, I proposed experiments with high schools through our teaching office, and they said that that didn't seem feasible. But in the second time around when I got it, I took that out and I just focused on course development and pedagogy, and that was more well received. And so then I wanted to end with building a career, so what's next? So I learned after getting this that funding begets more funding. So within the CSU, there are opportunities for matching funds, and if you have an external grant, it counts as a cash match. So for the Agricultural Research Institute, I was able to get a grant for iron and nickel catalysts to mitigate denitrification in agricultural pollutants for $83,000. Some things I'm looking forward to applying for in the future are a center grant for crystallography, other instrumentation grants, and more immediately is looking at international collaborations and a sabbatical. So my master's student is actually going to be traveling to Germany in December to do funding with the DAAD, which is the German, one of the German funding agencies. And so I'm hoping to leverage that for sabbatical work in Germany as well. And so to summarize, I just want to say preliminary results are absolutely key if you're going to apply for a career. Reiterate important components. Don't be afraid to feel like you're repeating yourself. Really focus on feasibility for PUIs. So in the proposal, I even specifically write students' names, what year they were, that they're an undergrad, and I underline it and bold it and highlight it to make sure people really see that undergrads are doing this work. And build collaborations. So start building that network well before you think you want to apply. And so with that, again, I mentioned I can't make the Q&A, unfortunately, but you can email me. My email is at the bottom. I have Twitter. You can tweet me or DM me. And we also have a group website, so I encourage you to take a look. Thank you for joining, and I wish you all the best. The next speaker I'd like to introduce is Cheryl van Buskirk. She's from CSUN, and she'll be presenting the next section.
Thank you, Chantal. That was a great talk. She gave a lot of points that I was going to do, but then hopefully I still have some complementary things to tell you about. So I'm at Cal State Northridge. I'm in the last year of my career grant, so I'm at the other end from Chantal right now. And what I'd like to tell you today is how my career award helped me fund a course-based undergraduate research experience that was well integrated with my research program in sleep. And the course was called FIRE, for Full Immersion Research Experience. And the research question that I'm focusing on is why we sleep. That is, what is the cellular function of sleep? Why do you get tired at the end of the day? Sleep is influenced by two major components, not only the need-based, but also the timing-based. So the circadian system controls sleep in addition to many other things. So I wanted to choose an organism that would help me understand why we sleep, and therefore I wanted something that isn't limited when it can sleep. And so in the nematode C. elegans, which I'll show you on the next slide, even though they have circadian rhythm, their sleep is not under circadian control. And so this means that we're only going to see the sleep when they need it. And so this next slide is going to show you when they sleep. So one time that they sleep is during development, and the other time that they sleep is following exposure to damaging conditions. And we know that they're not hurt, they're just sleeping, because as you can see, when I touch it, it moves normally. It's quickly reversible. And so these guys aren't just sick, they're actually asleep. And so one of the focuses of my research program is to understand genetic pathways, the cells, and the conditions that cause a need for sleep, this sleep need. Okay? And so about 2015, I wanted to write an NSF CAREER grant to try and integrate this research program with a course that I had taught once. And this was a course called FIRE, and I had redesigned FIRE from a previous version so that it was aligned completely with my research experience. And it went very well. So I had some preliminary data. One of the students, a minority student, went off into graduate school. Lots of them presented at posters at a symposium. And so Chantal was telling you about the importance of having preliminary data. That applies not just to your research component, but also to your education component. Start thinking ahead of time, a couple years ahead of time. What do you want the preliminary data for your research and your education components to look like? And start working on those things as soon as you can. And so just to give you an idea, in a 15-page strategy for the CAREER Award, I did 10 pages of research and 5 pages of education. So it wasn't an afterthought. And it was very integrated. And I even drew them a schematic to show them how integrated it would be. So you can see in this schematic on the right here, we've got, I'm teaching FIRE. This is producing not only data from the students, but it's also producing well-trained researchers that can then join my lab and continue to work. They can go to meetings and present. They contribute to publications. And that new research can then feed back in to the next offering of FIRE. So this is a very authentic integration. And I had preliminary data from my first one that really helped support the idea that this was going to work in the context of my research program. Here's just a few pictures of FIRE Lab. You can see that it's a pretty small group. It's only about 15 students, which is why it really needs funding, because as you know, the really small classes are hard to offer frequently. It's a reading intensive, writing intensive class. I don't want more than about 16 students in there. And so without funding, it's very hard for me to be able to teach that on a regular basis. I usually teach it in the fall, and then the students can attend an on-campus symposium in the spring called CSUN Symposium. And because it's a senior course, then they tend to graduate the next semester. And if any of them want to stick around, this is a really well-trained group. They can join my lab as research associates, supported by the NSF grant. And so it's a very nice situation for me and for them. We always have a good time at the symposium. They also contribute to major publications from my lab, either in the acknowledgements section or as authors. And I wanted to just point out a really good way to get your students to be first authors on papers. And so this is, if you haven't heard of it, there's a few of these popping up, but my favorite is micropublication biology. This is published by Caltech, supported by the NIH, indexed in PubMed, data curated, peer-reviewed, and it's a very rapid publication. And it's a great way to have your students who are doing solid work that doesn't fit into a larger story, get that out there and get practice in writing and getting that first author paper on their CV. That's one of my favorite things to be able to get my students to do. So FIRE can be a lot of work to teach because it's never the same twice. That's also the reason why it is an authentic research experience and it's more fun to teach. So for example, one semester we could do a mutagenesis screen to look for sleep defective mutants. Then we'd take some of those to my lab, work on them, pare them down to what we think are the most interesting, most penetrant ones. And then in the next semester, the students could do a higher level characterization, some genetic mapping to find out where it is, select their favorites, and send them for a whole genome sequencing. That's probably the end of the semester. So we'd select our favorite few, bring those back to FIRE the next time with those genome sequences, and then have the students from that try to find which candidate their gene is, right? And it's a really good experience for them to see that sequencing isn't everything. You actually have to have that mapping data from the previous round of FIRE as well. Okay, and so that's a really good way for our lab over the course of time to accumulate a lot of really interesting mutants. And one of these is a mutation that is defective in the development of a particular neuron. And this neuron is called the ALA. And it's a neuron that apparently is a sleep-inducing neuron. And so this is pretty exciting for us to find out. And through our genetic screens, not only have we found out the tissue that is important for sleep, but also this genetic pathway. So within this neuron, you require components of epidermal growth factor signaling. You require things like neuropeptide release. And with this array of mutants, what we can find is that if you're sleepless after exposure to damage, you're not going to recover the same as the sleeping animals do. And so this black line is the wild type animals. They got to go to sleep and they're going to survive very well 4 days out after a severe heat shock. 90% of them are still alive. But if you take a look at this blue line, these guys don't have the ALA neuron and they're not surviving. And same with these guys who are missing one of these signaling components. The exciting thing about this finding is that this was first observed by a student doing a hypothesis-driven project within FIRE. So this was my favorite moment in science that I can think of in the last 5 years, I think. And I've had a lot of really fun moments. Okay. So this led us to the hypothesis that cellular repair actually can limit sleep duration. Another way of saying that is cellular damage is giving sleep pressure. If that's true, then what you could imagine is that if you have a mutant that can't repair very well, that it would need to sleep more. And so we tested this in the context of UV-induced sleep. So we already knew that if we hit them with ultraviolet light, they're going to go to sleep. And when we know all the things that you can hit them with because the FIRE students got to go crazy doing all of these things that are kind of torture to worms, but don't worry, they only have 1,000 cells, so it's probably okay. And they figured out all of these things that would put the worms to sleep, one of which was ultraviolet light. Now, there's an important protein called XPA1 that's required for nucleotide excision repair. And so we hypothesized that if these mutants couldn't repair their DNA very well, they might need to sleep even longer following UV exposure. And this is exactly what we saw. So if you look at this graph on the left, you'll see the black line is a regular wild-type animal. They sleep for about 7 hours following UV exposure. These XPA1 mutants who don't repair their DNA, they sleep much longer. And then we know that this extra sleep isn't just them being hurt, because if you cross that into a mutation that lacks, a mutant that lacks that ALA neuron, you see that sleep disappear. Okay? So these are pretty doable experiments by these students. They are making some genetic crosses, they're analyzing behavior, they're looking at sleep versus not, and they're creating wonderful, wonderful projects in the lab. Lessons learned. It is still challenging for many students to do worm genetics. Physically, it's difficult because the worms are too small, and genetically, conceptually, a lot of them aren't as prepared as they could be. I found that teaching a cure takes a lot of time because it's different every semester. But on the bright side, it's not time that's taken away from research productivity because it's authentically contributing to your research program. And so in general, I think having an NSF-supported cure is an excellent way to really enhance your research productivity. Future plans is to help prepare students better by creating a pipeline cure, taking some of those prerequisite courses, adding in some modules that's going to help prepare them for the types of things that we do in FIRE. And of course, this would be in collaboration with other faculty in the department, and we're starting to do that now, and that's pretty exciting. Since I'm very close to the end of my career award, it's time to think about the next steps and spreading FIRE and looking for funding to expand this paradigm so that we can offer this course to even greater numbers of students. So in summary, research-wise, it looks like cellular damage might be one of the things that's a major component of sleep needs, not just in worms, but they're starting to discover in other organisms as well that this might be a very deeply conserved phenomenon. For NSF CAREER, the education and research components really need to be mutually beneficial for your proposal to be outstanding. Chantal was telling you how everything is an excellent one, but to make it truly outstanding, it should be an authentic integration, and your research should benefit from your teaching and vice versa. All right, so I'll be around here for the Q&A as well. Our next speaker is Dr. Kim Blisniuk from San Jose State University.
Hi, everyone. I'm an earthquake geologist, and today I'm going to present to you a brief summary of how geology is used in assessing seismic hazard and how this relates to my career grant. What is the role of geology in hazard assessment? Geology provides deformation models, and these models are actually first order models that go into earthquake hazard and earthquake probabilities. And these types of studies are not just important for earthquake geologists like myself, but they're important for all the residents and people that live in California or in areas that are earthquake prone. The figure to your right shows the San Andreas Fault. And as you can see, the San Andreas Fault is the boundary between the North American plate and the Pacific plate. And it starts in Mexico, and it goes and it cuts all the way through California, up through the Mendocino Triple Junction. And all of the communities, universities along this fault and all the faults in California are affected by earthquakes. And so in this presentation, I'm just going to give you why understanding deformation on these faults are important and how we can use, how I use this to leverage my career grant. The deformation models are a function of what we call fault slip rates. And what I've shown here in the figure to the right, we saw where the fault cuts up through the state of California. And here what I'm showing is the fault in Southern California. And there are three main faults in Southern California. There's the San Andreas Fault, the San Jacinto Fault, and the Elsinore Fault. And depending on which CSU you're at in Southern California, you probably know which fault will produce an earthquake. And the most important aspect of assessing earthquake hazard is something called a fault slip rate. And a fault slip rate is directly proportional to a fault hazard potential. And that's because the faster a fault slips, included with the most recent time the fault ruptured, tells us how likely the fault will rupture. And so these lip rates are only observed through what we call geologic data, observing how the landforms across all of these faults and along all of these faults have moved over time. And that's what geologists like myself do to look at the rates at which these faults move. And we always hear about the big one in California. And that big one is supposed to occur on the San Andreas Fault. And the San Andreas Fault last ruptured in 1857 towards the north, all the way down, and it ends around Palmdale. And we always hear about the big one happening. And that big one is supposed to occur any day now. And knowing when and where this big earthquake is going to occur is one of the big questions in earthquake geology. And we always think we know it's going to happen, and we know that because earthquake geologists have recorded that the last earthquake to occur on the southern San Andreas occurred around 1690. And since it occurred around 1690, it's almost 300 years overdue for a large earthquake. And so what I'm going to show you next is how an earthquake geologist like myself can look at the landform and use this data to better understand how the fault is moving over time. And so what you see here in this figure is a fault called the Mission Creek Fault. And the Mission Creek Fault is a strand of the San Andreas Fault in Southern California that has not ruptured in historic time. And what you'll also see is a blue polygon or shape. And that blue shape is the modern river of what we call Pushawalla Canyon. The location of this site is in the Coachella Valley. And what you'll also see is that this modern channel has 3 other channels that no longer has a source. And these 3 channels you can see are channel 1, 2, and 3. And these 3 channels are offset by about 2 kilometers, 1 kilometer, and half a kilometer. And what we can do as earthquake geologists is we can use these landforms, we can date them with something called quaternary geochronology to obtain fault slip rates. And this fault, before I studied it, was not hazardous. It was not a fault that was going to produce a big earthquake in Southern California. But after working on this fault for my postdoc, I was able to show that this fault is likely the most hazardous fault in Southern California to rupture in an earthquake. And the slip rates are about 22 millimeters a year, which is half of the rate of the boundary between the Pacific and North American plate. And so using this data and knowing that what we understand about faults in Southern California may need a revision, and that some of the models that we think are true may not be true, I was able to leverage the work from my postdoc into a career grant. And in this career grant, what I showed is that we've known for over the past 30 or 40 years that hazard models in Southern California show that if the San Andreas Fault were to rupture, the likely scenario of an earthquake rupture was going to occur on a fault called the Banning Fault Strand. But in the work that I've been doing, what we see is that we may have missed information. We may not have observed all the geology where faults are rupturing through the surface. And if that's the case, I proposed a new model for hazard in Southern California, and what we call these kinematic models. And in this model, I was able to hypothesize that we believed for so long that there's going to be rupture on a particular fault, but if we actually take a closer look at these models, the data show that we can reevaluate what we know and what we understand about faults in Southern California, about earthquake hazard, and provide a different model where we can actually quantify the data. And this is really important because there's so much new, there are so many advances in the sciences all the time, and many of the models that we believe or we were taught may not be valid models given more and more data. And so I think my NSF career grant really was successful because not only do I include a lot of student training, but I was able to challenge pre-existing models of what we know in the science of earthquake hazard and earthquake evaluation. And so with those observations, I went back and looked at how these faults may be mapped in the area, and I can show that we may have missed not just where the active fault may be, but we may actually have unmapped structures. And so there's always still more science to do, even though there's always a way to, there's always models that are accepted, but we can always challenge these models. And these photographs on the right here just shows the sediment that have been offset along the fault, what we can observe in geologic and geomorphic data of offsets that are caused by surface rupturing earthquakes. And here, what you can see are these sediments that have cut through the surface, and it's only earthquakes that can cut through the surface that are very dangerous. Earthquakes are happening all the time. And so in the bottom picture, what you can see is an offset channel that has moved to the right. And that offset channel has moved to the right over a time period where you have multiple earthquakes. And we can see all of these observations in the landscape. And so what I do with my students is that we go out into the field and we map these faults, we collect these data, we go back into the lab and we process the chemistry of these rocks in the lab. My students then go to Lawrence Livermore National Labs or various labs in the San Francisco Bay Area to complete the work. And so not only do they get the experience of being in the field and being trained, they're also in the lab and doing work. So it's there's a multi-component to it. And so this is just my last slide showing that geology is critical for earthquake assessment, and we need to do it right. This is a logic tree of how earthquake probability models are made. And you can see geology is up high in this logic tree. These rates go into these models. Every fault has a slip rate on it. That slip rate is then transferred to forecast the likely percentage of when the next earthquake should occur. And so with that, I'm going to introduce the next speaker. Blake Riggs is from San Francisco State University, and he will talk to you about cell fate.
Thank you, Kim, and good afternoon, everyone. It's a pleasure to be here and talk about my research and talk about my application for the Career Award. And just to let you know, I'm at the end of my Career Award, so I am currently searching for new funding, but it's been a great ride. and tips as well along the way. So just an overview. Typically, you would list your aims and the aims I proposed, but I just wanted to give kind of an board study. I'm a cell biologist, and I've always been fascinated with this picture of mitosis. Now, a view of mitosis, and if you've taken any biology class, you're probably familiar with this, that there's phases of that going through division, and you'll see these beautiful chromosomes that they condense the mitosis on the metaphase plate. They are segregated at a phase and go into new daughter cells, and that's how you divide a cell. Now, the question I started my lab with, and the question I was really interested in, is, well, what about everything else in the cell? I mean, we know a lot about chromosomes, and a bunch of studies have shown that this is essential for cellular function. They show that if you have mistakes in inheriting chromosomes, this could lead to disastrous effects like aneuploidy, which is a hallmark of cancer. So, and this is the way I think of cell division. And while this picture is very difficult to see and don't focus necessarily on the technical details here, but there's a lot more going on in cell division in just chromosomes. So then I started to ask the question, well, how are other organelles inherited? Namely, thinking about the endoplasmic reticulum, or the ER, which is an essential organelle that is responsible for secretion of proteins, regulation of calcium, and general cellular homeostasis. And this, we started to link this inheritance of ER into a very essential process, which I did not see these two related until we found some really compelling data, with this idea of cell fate selection. So many of you are familiar with movies where you see embryo is fertilized and it starts to divide, and it goes to 2, then to 4, then to 16, then to 32. And you've seen these divisions, and you've seen these movies in your science classes. But the question that I'm trying to answer with my research is really, how does that process happen? How does a cell just get its job and adopt its fate? And we find that this actually happens through an asymmetric cell division, and that this asymmetric cell division, one cell gets more necessary components than the other. So in studying this, I study this in fruit flies, and fruit flies are the first model organism. They're great because they're very inexpensive to work with. They have a tremendous amount of genetic tools. You can do approaches like biochemistry with them, but you can also do really high-end imaging of cell division, and that's what I'm really interested in. And in doing this, we started to ask the question, well, what does the ER, the endoplasmic reticulum, this essential organelle, look like during cell division? Now, I do want to mention, oh, there's the dreaded document showing the break there, but I have the movie set up. I'm going to show you guys a movie of what we're talking about. But what I want to, before I play the movie, I just want to say, I applied for the Career Award, and I applied for it 3 times. So, and I got it on the last try. And I think one of the lessons that I want to impress on all of you is that as we are scientists, we don't give up. And I think it's important that when you apply for these awards, you keep applying and you keep learning from your applications and making them better. And that's kind of one of my lessons I want to impart on you. Now, shown here in this movie that I'm going to play was a discovery that we made in the lab. And we made this discovery on like my 2nd attempt at the Career Award. And so in this, we are showing just general cell division during embryo development. And in this figure here, hopefully you can see my mouse, that in this figure here, we're looking at cells that are dividing in the anterior portion of the embryo. And what you're going to see is that one cell is going to get more ER than the other one. So in green is the ER, and in red is the DNA. And hopefully this will play, and you'll watch, and you'll see that one cell will divide, and it'll just get more ER than the other cell. Well, when we were looking at this, we were just asking the very basic question of how the ER is inherited, and we quantitated this, we looked at it. But what we started to realize is that this asymmetry is very reminiscent of mechanisms that are involved with cell fate decisions. And actually identified a protein that is responsible for this asymmetric division, and this protein is known as jagonal. And it's, if those of you who speak Korean may, it means for a tiny egg. But what we found is jagonal plays a role in oocyte development, but we found another role for it where it actually is involved with ER inheritance. And you can see in this next movie I'm going to show you, we're filming that same shot there, but now we're looking at an ER, at a embryo that we've gotten rid of jagonal, so we've knocked it down. And shown here in the movie, again, green is the ER, and red is the DNA. And let's see if it'll play. And we're going to look at the same thing, and instead now these cells are dividing much slower, and there's no asymmetry. You can see one's going to try to be asymmetric, and then it snaps back. And so we found that this protein jagonal is responsible for this ER asymmetry, and we were able to publish this. Now, and some other bits of the story is that we were able to show that jagonal also, this ER-associated protein, is also asymmetric. And the link was we found that it really played a role in the cell fate selection of neuroblasts. So these stem cells that in Drosophila and in humans also, that now give rise to neurons and ganglion mother cells, so development of the brain. So there we've linked this idea of an organelle playing a role in cell fate selection. And this is a lot of work that I did during my career award. And I do want to mention, and I'll show this again later, that this was done by undergraduates and master's level students in my lab. Now, where we're going with this, and this is really preliminary data that's just come out of my lab and is going to be the basis of my next grant that I'm submitting, hopefully soon, is that we see that when we get rid of jagonal, we knock it down, we look at mutants, that there's an uptick in stem cells. So we shift cell fate selection back to a what's called a proneural state, or we increase its stemness. So this is now giving a direct role, showing that jagonal plays a role in cell fate selection. And there's a lot of quantitation in this, but the most important thing you will see here is this in red is a marker known as dead pan, and this labels stem cells. So in jagonal mutants, you can see all the red shown there, and those are cells that have turned into basically a stem cell. And this is now going to be the next wave of what we're going to look at here. So with the Career Award, and NSF is really purposeful in that you need to be both a scholar, but also you have to be, you have to have broader impacts. And we've seen people identify their broader impacts. What I realized and what I looked at is retention of students. And I thought about my own, how did I make it? How was I retained? How did I make it to get my PhD? And I looked at, it wasn't necessarily that were there for me. It was rather my own peers. And I created a near peer mentoring program. And many times we think underrepresented students in the sciences? Well, we don't want to burden them with community work. We don't want to lay on them. We just need them to focus on classes. I disagree. What I found in talking with students is that they want to with community. So what I did is that a near mentor that they were able to do elementary science days. So 4 times a year, we would invite elementary students to the university and their parents. We do STEM activities here. We worked with local chapters. One of them you see wearing the SACNA shirt. We worked with the SACNA chapter. We also did outreach with our students with Dinner with a Scientist, where they went to the local elementary school and talked about their journey in science. Now, one thing in doing this, the students also expressed, especially our African American students and Black students, were like, we want our own group. So we created a group known as Black Excellence in STEM, or BSTEM. And these were outlined in my broader impacts, but I didn't really talk about creating this group because I didn't know I would. This was actually research that we did. And from my broader impacts, we now created this Black Excellence in STEM that is important with doing community service and working with the community. So what I just want to outline is that some of the lessons learned, and I'm going to pass it on to our next speaker, is that, again, you have to organize, outline yourself, learn when to change course and let go of some projects, practice empathy with students. I believe in growth mindset, that everyone can do science, and remember who you are and why you're here, and never give up. Okay, with the next steps, we are going to, again, try to answer these questions of how the ER plays in self-fate selection and learning course in STEM mentoring. Okay, with this, I am going to pass it on to our next speaker. This is the lab that did the work here in the right. And I want to introduce, I have the pleasure of introducing Geoffrey Lovelace from Cal State University, Fullerton.
Well, hi everyone. I'm really grateful for the opportunity to talk with you all today. And I always just so blown away by all the amazing work going on in the CSU. It makes me really proud to be a part of Cal State, to see all this amazing work that our students and my colleagues are doing. I'm going to talk a bit about computational gravitational wave physics and astronomy at Cal State Fullerton. So I'm one of 4 faculty members in the Nicholas and Lee Begovich Center for Gravitational Wave Physics and Astronomy at Cal State Fullerton. And our mission is to do research, education, and outreach in gravitational wave science. We have about 25 students now, and about 33 students have come through the program. And my group in particular focuses on computational gravitational wave science. And so first, I wanted to say something about what gravitational waves are. And I've got a little movie here that's going to show up where that broken document is at some point. I see it over on the side here, but I can't move it myself. So there are 3 components to our 3 components to the program. One is we're doing supercomputer calculations to crash black holes and neutron stars into each other. And to predict the gravitational waves that come out. So here's a little movie showing you how that would work. So spiraling around each other, and they're faster and faster until they smash into each other and make a new big black hole. And as they do, they send out these ripples of curved space and time at the speed of light out into the universe. So our calculations are helping to model what those gravitational waves look like or sound like. It's not really sight or sound, it's a new sense that humans have. But the calculations help us interpret what we see. They help us reconstruct what the curved spacetime of the black holes was doing right when they collided. We also have been doing some work modeling one of the important sources of noise in gravitational wave detectors, the fact that the mirrors the detectors use have a temperature. And the educational component is a 1-week summer workshop that I'll tell you a bit about on the next slide. So when we model merging black holes and neutron stars, we're part of a collaboration called the Simulating Extreme Spacetimes Collaboration. And we use a code called the Spectral Einstein Code and big computers to solve Einstein's equations of relativity to model the curved space and time as black holes crash into each other. we have to use big computers because there's no approximations that anyone that works when the space-time gets so distorted. And the space-time can get strongly enough distorted. Let me show you another movie here to show you in about 100 times slow motion what it would look like to have two black holes orbiting and merging. The gravity is so strong. I guess I better see if I can get this to play here. The gravity is so strong that it bends the background starlight like a lens in this sort of wibbly-wobbly pattern. Here, I'll see if I can play that one more time. So we want to connect that kind of turbulent souls that travel across the universe and that our detectors pick up. When we model thermal noise, it turns out we can use very similar techniques to model the behavior of mirrors in LIGO's detectors. So here's a picture on the right here of half of a cylindrical mirror modeled on our computer. For the workshop, what we've been doing is it's an annual 1-week workshop where we invite 20 students from a community college we're partnering with, Citrus College, and they come with no prior experience and get introduced to things like orders of magnitude in computing and realizing just how big supercomputers get, how they can carry out more mathematical operations every second than if you had every human who ever lived doing computation for you. They also get an introduction to programming with Python, and they learn how to run our spectral Einstein code to do a simple case of two black holes smashing into each other head-on. And students in my research group are in the middle of this. They come in and talk about their research and lead a panel discussion talking about transferring to a four-year institution and then thinking about grad school and careers. So some of the results that I wanted to highlight, our students have contributed to the Simulating Extreme Spacetime Collaboration's catalog of gravitational waves. Black holes come in different sizes and shapes, they spin at different rates, and all those things change the gravitational waves that get emitted. The waves are like a fingerprint, and if you know that fingerprint, you can figure out what it was that you saw. Was one black hole bigger than the other, or how fast were they spinning? So here's a movie showing the first 10 colliding black holes that LIGO observed. These are supercomputer simulations of those first 10 black holes. And the different black holes lead to different patterns of the waves on the bottom there. So like here's a different case where one black hole is much smaller than the other, and then the fingerprint turns out to be different. Now, I'd like to make this a little smaller, please, so I can see the figure underneath, but we'll let this keep playing. Another result that we were able to do is our group is specialized in simulations where the black holes are spinning nearly as fast as theoretically they're able to. And one question that I had was, if there were such a thing out in the real universe, would our detectors recognize it? Would they be able to successfully decode that the black holes were spinning really fast? I talked to one of my collaborators, now a professor at Caltech, and I'll apologize in advance for struggling to pronounce her last name, but Katerina Chatziano at Caltech is an expert at taking model waveforms like the kind that we produce, and one of the things that she knows how to do is how to make a simulated gravitational wave event from these computer calculations and then analyze them just like a real event. So this plot on the bottom is plotting how fast a big black hole is spinning on the bottom and how fast the smaller black hole is spinning on the top and on vertical axis. The little x's are the true values that we hid in simulated detector data. When both black holes are spinning the same way, both arrows are up or both are down, that means they're both spinning the same direction, then our methods do pretty well at figuring out what happened. But when one black hole is spinning clockwise and one is counterclockwise, when one arrow is up and one arrow is down, the x's can be very far off from these little contours in the middle, and they miss it. So that's one thing that we learned. In the thermal noise project, we learned that it turns out if you coat our mirrors with a crystal, it's harder to model the thermal noise in that case, but the actual result that you get compared to if you just used a glass coating instead, like we usually do, turns out it doesn't. The model isn't terribly sensitive to that kind of difference. Now, as far as the workshop goes, we held one in 2018 and one in 2019, and we were going to hold one this year, but unfortunately, COVID stopped us from being able to do it. But I would say one outcome I wanted to highlight is a participant in our 2019 workshop, Marlo Morales, transferred to Cal State Fullerton and is now a member in my research group, working on some of the work that I'll tell you about in a little bit with next steps on the last slide. Things didn't go exactly the way I thought they would when I wrote the career proposal. In particular, I wanted to get into not just modeling black holes crashing into each other, but having a black hole tear apart a neutron star, like the picture you see here, where its gravity is so strong it takes a nice little round circular neutron star. It'd be spherical, look like a circle in this picture, and tear it apart in this messy pattern. But these simulations turned out to be a lot more technically involved and need a lot of fine-tuning that we didn't expect. And this led us to change directions and start contributing to a brand new code that isn't delivering science results yet. But having a career award gives you some extra time to invest in working towards something new and helping to get it to the point where it'll be generating science. I also didn't get the career award the first time I applied to it. I definitely endorse the suggestion from some of my predecessors in this webinar that it's good to get a track record of funding first. And at the NSF, the Research and Undergrad Institutions, or RUI, is definitely the way to go. And advice that I have about the application process, I would say the research component has to stand out. It has to be a little more. It has to have some sparkle to it, and sometimes I can be a little self-deprecating or hard on myself, and writing a proposal is not the time to do that. You want to show why what you're doing is cool. But also, I would say play to the strengths that the Cal States have. We are models of integrating research and education, and that's exactly what this award is about. Neglecting the educational component in favor of research is something you definitely don't want to do. So I have one more video I can show you for our future plans. So we're continuing to develop this brand new code, and it's not. And just recently we were able to get the first pair of black holes orbiting each other in this new supercomputer code. So I'll play this movie for you here. What you're seeing in color is how gravity is so strong near the black holes that time is ticking only half as fast as it is far away. One thing that makes this code new and why we have to write things from the ground up is the parallelization works differently to help it scale to the computers we'll have in the next decade. It's not just the computers we have today, so run on 100,000 or 1 million processors. So the big difference is our old codes, the spectral Einstein code, uses a kind of parallelism called a simple parallelism. And this is sort of like, imagine if you're on Zoom, everyone's trying to work together, then you ask someone a question and they weren't paying attention, and then they start talking and they're muted and everyone has to wait, and that's no fun. And the task-based parallelism that our new code uses is more like working on Slack or with text messages. When you have a question, you send a message and then you go on about your business, and whenever you get a reply, then you can react to it. So it's more reactive like that. And getting ready to help get this code producing science results, and then using that to interpret the results from future gravitational-wave detectors is our big long-term plan. So that's what I wanted to tell you about. And here are just some pictures that some of the students have done here. They've traveled to conferences. They've talked to the students in our workshop about the research. And the top right is one of our students inside one of the arms of one of our gravitational-wave detectors. So next, I'd like to introduce Matthew Povich from Cal Poly Pomona, and I'll just turn it over to Matthew.
I just wanted to say it's, Jeff's always a tough act to follow. He can say he's self-deprecating, but the research he does is sort of self-evidently very cool and has won Nobel Prizes already. So I'm going to talk about sort of a more humble topic in astronomy, but I think a really fundamental one. And my presentation is going to focus as much as possible on the students and collaborators who I've had working with me. So On the original slide, I had bolded some of the students. That seems to have been lost in this version, but that's okay. I will get to them later. So for my career award, which just ended in June, the basics here for intellectual merit and broader impacts are written down. I'm an observational astronomer, so I use lots of data and surveys that are obtained from all over the place. It's actually good to be it's a good project for a CSU because I don't need access to a big fancy telescope like you'd get at a research university. I've become something of an expert in just downloading data off the internet and doing specialized analysis and being parts of large collaborations, so I can get access to really the best data that I need for my sort of work. And the basic problem here is the question of how alive is a galaxy like ours. You have galaxies make stars. Stars form when giant interstellar gas clouds collapse and break apart under the influence of gravity. When stars form, you get planets along with that, which we should all care about because that's what we live on, a planet going around a star. But there's quite a bit of uncertainty about sort of how do you measure this level of activity, which is quite fundamental in our galaxy and others for the way the universe works. So that's what this business of calibration of galactic star formation rate indicators means. It's like, what are the tricks astronomers use to say how much star birth is happening in the universe and how well do we know that? And then for the broader impacts, I have some boilerplate here, but as we all know, being CSU people, I'm working with not the usual suspects, students who are members of underrepresented minority groups, first in their family ever to go to college, not typical sort of well-to-do people who maybe end up populating a lot of PhD programs. Ultimately, you kind of get that automatically being a professor at one of our campuses. So as the outcomes here, I've listed off things, and like I said, I'm most proud of the things that my students have done, so let me get to some of that. This is a marquee project that I've been involved in for a decade and ended up having to take over leadership of during my career award. This was not foreseen. I was supposed to have a collaborator who was leading this project. He was able to leverage his skills working on this and other projects to a very lucrative position as a programmer at Google. So he left the project. I took over science leadership. And then an undergraduate student from Sri Lanka named Tarindu Jayasinghe sort of just dropped into my research group and reinvented basically all of the code and data analysis that this former postdoc, now Google programmer, had worked on previously, which was kind of a miracle. But Tarindu is one of these unicorn students who is unbelievably brilliant and works like a machine. So this example of Tarindu's work was published in 2019. We basically took images of a huge swath of the sky, basically most of the star-forming real estate in our galaxy. What I'm showing here is really just like 1% of the data that we analyzed. We put them up on the internet, and then regular people all over the world, thousands upon thousands of them in hundreds of countries, were able to go and draw these white ellipses. Now, they wouldn't be seeing a big complicated image like what I'm showing here. They'd be seeing a little piece. And then from all the pieces, we put it together and stitch what you see up here. This is the raw data. Then Tarindu wrote this code that could sort of analyze where do you have a whole bunch of white ellipses overlapping each other. And that's the second thing. We call these a cluster. And then once you've got a cluster, it seems to outline what we call a bubble, which is this sort of yellowish, reddish-green rim in space. It's a particular shape of a nebula. And then you do a smart weighted average of all these different ellipses, and you get this final catalog, which outlines these structures that we see everywhere in space. And we ended up cataloging 2,600 of these, which were based on this input. And there are some other objects we cataloged as well, something called a Bow-Shock Nebula, which I won't show, but they're interesting because they help us identify brand new massive stars that no one ever saw before, and those things are rare and powerful. Well, another example of a really high-level student project, we're still wrapping this up, finishing up the paper. A student named Evan Nunez, he analyzed the Carina Nebula, and we had this set of stars that we found in X-rays and infrared images, and there's several hundred little multicolored symbols on this image of the sky. It's a pretty big image. You could fit, I think, like 4 or 8 full moons inside this image. And this was a sort of niche project to talk about the X-ray flaring properties of the stars that we see in this image, and a neglected thing where the stars are relatively massive. They're at least, the ones we're looking for, at least 2 times the mass of our sun. And stars like that, they're when most of their lives are quite different from the sun. They don't have magnetic fields and sunspots, and they don't make flares, and they shouldn't make X-rays. But it turns out when they're young, they actually do. And Evan's paper is kind of definitively showing that this happens. So here's a plot he's made here of what's called a luminosity function and these funny units. And how powerful these things are in X-rays. And the main takeaway here is that the green curve is shifted to the right compared to most of the other curves, and then the red one's in between. And what that tells you is that these things that we call IMPs are systematically more powerful in emitting X-rays than other types of young stars that we see in this nebula. So why would we care about that? Well, it turns out that because this red curve is intermediate, and we think the stars representing the red curve are ever so slightly older, meaning like maybe 1 million years older or something, which is nothing in astronomy. That's like 1 million years like that in astronomy. We can use these sorts of things to help help calibrate time scales for star birth. And like I said, we want to figure out things like how quickly are stars forming in our galaxy and elsewhere. One of the hardest things to do is get a handle on your time scales. So this really novel measurement and work that Evan has done is really showing that this can be done the way we want to do it. So then this kind of, then I can take this and synthesize similar work for all of these. There's like 18 different nebulae in here. And what I'm plotting is this time scale on the x-axis in millions of years. Like I said, that's the quick, I know it's funny, quick time for astronomy, for things to happen. And on the y-axis is a measure of infrared brightness. So that tells you, like those bubbles I was showing in the first slide, all those nebulae that Terindu and company were finding in the Milky Way project. The brighter they are, the higher up they would be on this plot. And the correlation here is actually much better than I thought it would be at first. It's a pretty steep drop-off that I can fit with an exponential decay function. So what we're basically seeing is that the longer the stars have been existing, or the longer they've been forming inside one of these star-forming nebulae, the fainter the nebula gets. And we actually find that it drops off, by a factor of 2 in less than 2 million years. This turns out to be quick. The sorts of tricks astronomers use to measure star formation activity elsewhere in the universe, they won't tell you up front, what time scales are assuming, but it's longer than this. It could be longer than this by factors of 3, 5, or even 10. So this, I believe, would mean that we run the danger of underestimating how fast stars are actually being born elsewhere in the universe by these similar factors of 2, 3, maybe even more in the worst case scenario. So hopefully that's something that's worth doing in a career award, trying to nibble around the edges of how we understand the universe by analyzing data in novel ways. So here are some pictures of my group. So the Terindu above mentioned student, he's on the right in the picture on the left. He is standing with Don Dixon, another student of mine. They were classmates in 2017, and they were the first and second authors on the Milky Way project paper. They're pictured here with Chris Lintott, who is the principal investigator of this entire platform called the Zooniverse, which has had over 100 different science projects and counting put online. So he leads the whole thing. He's out of Oxford, so he's kind of well known in England the way Sean Carroll is here, or Neil deGrasse Tyson. So he's posing with them at the conference. Evan, I talked about it here. We're on a hilltop in England on the right, and that was his first trip ever out of the country. And now Evan is a PhD student at Caltech, the first Cal Poly student to go there in astronomy. I mean, it's like a top program in the whole world, and they don't take very many students, so that was quite an exciting achievement. And Evan has just passed his qualifying exams, so I'm super proud of him. And then two others of my students, Jessica and Jackie, are here in the middle. I didn't get to mention their work explicitly, but they're also, Jessica has been on several of my papers published, and Jackie is my most recent student who's done work that is going to be published, well, as soon as I get time. Quick thing about my outreach, because I'm over time. Other than this whole citizen science world-spanning kind of activity, we do local outreach at schools in the Pomona and LA area. We have gone to dozens of schools and reached thousands of people giving astronomy talks and setting up telescopes. And in none of these pictures is it me giving the talk or doing the telescope, although I do that sometime. I'd like to give a shout out in the upper left. That's Brianna Binder, who I think is here on the meeting. She was a postdoc working with me on this grant, and she is now faculty at Cal Poly. And you know, she presented a couple times at this program just to help me out, but she's doing her own research now and all sorts of cool stuff. All right, let's see, one last thing. So yeah, so this is just my future plans. Right now I'm just teaching online like everybody, and that's all I can do to stay afloat with that. But when I can do that, I will I will read Evan's paper and make sure it can get submitted, and after that I'll do my own. And I'm still applying for grants and things like that, and maybe in the quick Q&A session I can give any words of wisdom that people may want. But you can see on here some of the words of wisdom that I'd like to put forward. So my time is over, and I want to introduce our next speaker, who is Nina Robeson from Cal State Fullerton. So hi, Nina, and bye, everybody.
Thank you, Matthew. Thanks, everybody, for being here. Thank you for your interest. I'm a mechanical engineer, and so I will talk about some highlights from my career award that I received 2 years ago, which is on robust geometric design of mechanisms for interaction with uncertain environments. What is the problem? Many labor-intensive manual tasks that are common in industry have not been automated so far due to high level of deformations within the objects that need to be manipulated, or variations and uncertainties within the operational environment. And so some examples over here you can see on the pictures are produce picking and harvesting, also sorting potatoes, for example, on a conveyor belt, sorting good potatoes from rotten potatoes, from debris, from stones, and so on and so forth. So similar cases exist in other labor-intensive industries, such as construction industry, hospitality industry, healthcare industry, manufacturing, to name a few. And so the goal of my career proposal is to advance basic knowledge on human-robot interaction and environment uncertainties, and incorporate some of those conditions on interaction and uncertainties on mechanical design level, leading to hopefully future novel hybrid design control frameworks and the creation of next generation user-friendly robotic devices that will be working alongside with us, with humans in our workspaces of the future. So the expected societal outcomes include transforming the health, safety, and quality of work life for future workforce performing manual labor-intensive tasks, and also, of course, increasing U.S. productivity and economic growth. The proposed design framework is expected to be assessed by solving a number of industry-related problems in the healthcare, biomedical, and agricultural areas, and of course engaging students throughout the way in the development, in the validation, as well as within the dissemination process. The educational and outreach activities of the career proposal, the outreach goal is to prepare the next generation of engineers of a broad diversity through emphasizing early cross-disciplinary research experiences, as well as innovation and entrepreneurship within the courses, the first and second year classes. Also, outreach activities include middle high school summer outreach program and another program geared towards increasing students' interest in research and assisting them in pursuing advanced graduate degrees. Some highlights for the past 2 years include the 1-week CSU Program for Education and Research in Biotechnology, or CSUPERB, summer course, where 2 of my undergraduate students, Juliana and Wesley, and I explored the potential for commercialization of the augmented reality wearable device that we were developing. Later on, we were awarded and advanced to go to an NSF I-Core workshop to continue exploring the RWET commercialization potential. In a bigger team, I would say, which we added to the team a business collaborator as well as a startup company owner. And the NSF I-Core program was about 3 months long. So it was pretty long. Other highlights from the past 2 years include some of my senior design teams from my senior design project course. You can see on the first photo, one of the teams was able to initiate the automation of some of the hazardous manual tasks for Bristol Industries. Bristol Industries is an aerospace manufacturing company located in Brea, California. Then the next picture is the patient walker team who worked in collaboration with a startup owner in Southern California. This team worked with the owner to design and manufacture a new intensive care unit to be launched on the market, and the team presented their work at the annual Southern California Conference for Undergraduate Research. You can see the team presenting at the conference. The last picture is the Edwards Life Sciences team and our industry collaborator in the middle, presenting our work on automating the labor-intensive manual sewing of a biomedical cloth process. The biomedical cloth is used in a heart valve replacement surgery, and they are presenting their work at the annual CSUPERB Symposium. At the end of the year, the student team was included in a joint CSUF Edwards Life Sciences patent application on automated sewing, which was a lot of fun for everybody. So since we are running a little late with time, I would just read some of the advice and tips that I thought might be interesting for you. So talk to industry, business partners, users of products, technologies, colleagues around to find out what the needs are and think how you and your students can address them. Apply for CSUPERB or NSF I-Core and go through the curriculum. It's very helpful and included within your career proposal that you went through that. Establish a lab from grants and startup funds. try to engage students in the research and generate some pilot data, publish some research papers on specifically the proposed career topic, propose a broad research agenda with high societal impact, and think of ways to show that it could be even broader. Prepare a summary of your proposed research and educational activities, send it to NSF program director, and schedule a meeting. Keep in mind that after talking to the NSF PD, you might have to tweak your idea to fit within the areas of interest. And don't be afraid to show that the research you're proposing is complex or ambitious, and you and your students would not be able to accomplish it on your own. Include partners in the proposal that are excited about collaborating with you on papers. Of course, they're not going to be funded from your career proposal. And lastly, schedule at least one additional meeting with the program director to discuss any concerns that you might have before the submission deadline. When discussing concerns, be honest and be open. And remember that NSF program directors are there to help you to be successful. And so, since my research is related to one of the NSF 10 big ideas for future investment, the future work at Human Technology Frontier, I just wanted to quote NSF with regard to the workspaces of future and the teams of future. The workspace of tomorrow is going to be a symbiotic coexistence, collaboration among humans, machines, and cyberspace. Interdisciplinary science and engineering research is needed to understand the benefits and risks of the new technologies and to enable the creation of these technologies with which humans will collaborate to enrich their lives in future workspaces. I'm really honored, and I am very privileged to be the advisor of one of those teams of the future that I called, that we call at Cal State Fullerton. And this team is the Titan rover team, which competes every year at the University Rover Challenge. The Titan rover team consists of students from all over across the Cal State, engineering students as well as science students working together. Yeah, Jennifer, would you please play, if we have time, I mean, you can play the clip. Thank you so much for listening.
Okay, great. Thank you, Nina. I know we are just at the end of our time, unfortunately. We can play just a little bit of it at the beginning, but I think we don't quite have time for the full 5 minutes because we do need to dive into the Q&A. So let's go ahead and start this, and we'll probably stop it a little bit early. Great. Thanks, Nina.
President and project lead for Titan Rover. Titan Rover is a two-part organization, an educational club and a URC design team with overlap between the two. The club's purpose is to inspire students to reach higher in space exploration and STEM education by using our past URC rovers and robotics demonstrations. The design team sets out to design and manufacture a complete rover for the URC. We now have 2 years of competition behind us and have learned some valuable lessons along the way. Last year, we developed Atlas from the ground up. This gave us a robust mechanical platform to work with, and we decided to make iterative changes this year and focus on the areas of opportunity we found through the analysis of our past performance. I am proud to introduce to you Calypso, based on the Atlas platform.
Beyond the lessons we learned from last year's competition, we decided to put the mechanical system through a series of rigorous tests to determine key locations for weight reduction to increase rover functionality. Testing provided valuable information, verifying that our system is robust enough to tackle any difficult terrain that is featured at competition.
Dual brush DC motors are mounted into each custom hub assembly in order to shield them from rocks. The tires have a custom tread design in order to increase our traversal abilities across sand, dirt, as well as steep slopes. We have prototyped and are currently testing a semi-active suspension system, which allows us to change the ride height and suspension settings at any time.
Using software provided by our sponsor, Altium, we are designing a PCB that will shut off any subsystem that is drawing too much current. In addition, we have a physical kill switch with dual circuit control using an internal solenoid. This allows us to physically cut all power to the rover, as well as send a remote command to depower the mobility system in the event of an undesired movement.
The robotic arm is designed for both form and function. It features 5 degrees of freedom and utilizes carbon fiber limbs that act as structural members while acting as an internal channel for all cabling and drive systems.
Feedback is designed into every joint of the robotic arm to enable real-time position of the arm and allow the use of inverse kinematics. The gripper can accommodate a vast array of objects using specialized gripper tips to maximize the points of contact on many standard and oddly shaped objects. The gripper can be field swapped for other types of end effectors if needed. The major joints and components of the arm have been designed with manufacturability and weight reduction in mind. Quick-release connectors for electrical and mechanical connections allow for easy serviceability during assembly and maintenance.
During the autonomous task, the rover is going to leverage the onboard processing cores in NVIDIA's Jetson TX2. We're utilizing both the GPU and the ARM processor on the computer vision system. We have built a custom model in TensorFlow for the tennis ball detection. Using machine learning, the model has accurately identified the tennis ball up to 16 feet away. Our navigation platform is based on a simultaneous localization and mapping model within the robotic operating system. Our sensors will construct an environment in which the rover will operate. After reaching the
Okay, we will consider that a little bit of a teaser. Thank you, Nina, for offering that video. We're just a little bit short and kind of coming into our Q&A segment, so I will hand it over to Frank to begin.
Thanks, Nina, and thanks everybody for your really wonderful presentations. It was really great. In fact, Nina, don't leave because it's time for Q&A, and we actually have one for you here. This is from a lot of these questions were obtained from the registrants. When is the most appropriate time to apply for an NSF career grant? Is it better to postpone it to the last years when you're tenure track or during the early part?
So I think that the best time for applying for a career award would be somewhere around your 3rd or 4th year as assistant professor. That would give you enough time the first 3-4 years to establish a lab from some smaller grants or from your startup funds, and also to probably decide what your career will be on, and then start publishing, maybe have a paper or two published on the subject of your career award, something related to it, as well as it will give you some time to just hire students working on the career award. So I think year 3 or 4 is the best. Also, year 3 or 4 will give you enough time to reapply. As you saw, most of us we hadn't gotten our career awards from the first time, so it needs, it needs some effort.
Great, thanks, Nina. I think this is a good question for Matt now. How did you convince NSF that you could actually pull off a career project, given you have a high teaching load in an undergrad-only department?
Yeah, well, I also had to apply more than once, so I didn't get that right the first time, I don't think. It's important to address those concerns and make sure you have institutional support out front and clearly articulated. So you need your department chair, you need your deans to be on board and supportive. You need them to be able to say things to you that you can pass along either back channels to the program director or diplomatically put within the proposal somewhere. Although it's harder to put these things in the proposal if it's career. If you're doing an RUI proposal, you have 4 or 5 pages to explain exactly how you're going to do the project and how it benefits the CSU undergraduate environment. But for career, you have to say, the broader impacts are rather profound, to do a project of this scale within the CSU. And then you to keep yourself sane, you want to work with your teaching scheduler, make sure that your course load is manageable, that you don't have too many new preps while you're doing the project, that you should, if at all possible, budget time in the budget for yourself in the grant to reduce your teaching load, and try to make that work from your campus. And I think those, though, now that we're seeing multiple career awards from certain campuses, and Fullerton, for example, is doing really well. Cal Poly has 2 now. It's probably easier when you can cite that they're. I was the first one at my campus, but if you can cite others who have done this successfully, it's easier.
Great, thanks, Matt. Let's bring in Geoffrey for this one. This is pretty much kind of a follow-up to the one posed to Matt. Do you believe it is more difficult to receive a career award at a CSU as compared to an R1 institution?
Yeah, that's a really interesting question. I think I would say no, and actually, I think the right way to look at it is not, oh, we've got something to apologize for, and oh, let's convince you that, oh, you might not think we're able to cut it, but we really can. I think it's the opposite. We can compete asymmetrically with people applying from R1 institutions. Sometimes R1 institutions focus so much on research that they're not as well equipped as we are to involve students, especially undergrad students, meaningfully in research. And what they really want from the Career Award is not a proposal that says this is the coolest research that will ever be done. They want something that integrates research and education, and Cal State as universities are perfectly positioned to do that.
Great, thanks, Geoffrey. Let's bring in Blake for this one. What failure have you experienced in your career, and how has this failure propelled you forward?
I think science is built upon failure, and I've experienced my fair share of it, especially with the Career Award. Whereas, I applied early on in my 3rd year, it took me 3 tries to get it. But the important thing, I think, is, like I said in my presentation, is not giving up on it, but also improving every time and learning from that failure. One of the things that I would encourage all of you to do is, when we get something like a grant rejected, we tend to have this this reaction to turtle, as I call it, like to go hide, throw, don't look at the reviews. But instead, I'd encourage you, go through those reviews, see what they're saying, call your program officer, ask them about what was the conversation in the room, what did they like, what didn't they like, how can I improve on it? They will really help you. Those program officers really are in your corner, especially at NSF. So I'd really encourage you to reach out to them.
Great, thanks, Blake. Next one's for Kim, and I know we've kind of chatted about this already in your presentations, but perhaps since you've served on several panels, what advice would you give, in addition to what we've already broached, to someone who's considering submitting a career or other NSF type of proposal?
I only know how it is with the Earth science or tectonics program, geology, but I would recommend, if you're interested in submitting a career award, to one, contact your program director. And I think Chantal may have mentioned this, but it's so important that your program director knows who you are and have an idea of what you want to do. Because oftentimes, at the end of the day, despite having a panel review, if they think your research and they think your broader impacts are strong and they believe in the work that you're going to do, they will fund your research. Serving on several panels, what I've learned is that try your best to have a strong, broader impact. Because a lot of the science that goes through the panel is very strong. I mean, you wouldn't be submitting an NSF proposal or a career proposal if you didn't believe in the work that you're doing and that the work is significant. And so oftentimes what I've seen, or what from my own experience, is that so many people focus on just the science, which is going to be strong, and they forget about the broader impacts and just devote like a page or half a page to it, rather than really focusing on how you're going to integrate your research or your knowledge to the broader community, whether it's working with local high schoolers or teachers or the community, but have that very well thought out, because oftentimes I've seen very strong proposals, and those proposals would have been funded if there was a stronger, broader impact. And being at a CSU, we have that as our leverage, and it's a strength because we serve a student population that represents most of California, and we can connect to these communities and to these student populations that not all universities are able to. And so that is a leverage. And so I agree that we are at an advantage when it comes to broader impacts and also intellectual merit, because we are doing great science at the CSU. And but again, broader impacts, which I think a lot of people forget about, and just calling up, emailing your program director. They are there to help you, and they want to see more faculty at the CSU funded, whether it's an NSF program or career grant. They really want to fund faculty at the CSU system.
Great, thank you, Kim. I think we are out of time, and so I'd like to firstly thank everybody in terms of your presentations. They were just actually superb. Speaker contact information is provided on the screen here. For more information on any of the presentations, please contact any of the speakers, as this could lead to a potential collaboration or proposal opportunity. In addition, a link to the webcast will soon be made available on the STEMnet website. Answers to questions submitted during the reg period and unanswered during the webcast will also be answered shortly. And as you can see on the PowerPoint, there's a pointer as to where they will be. In addition, we'll be emailing a feedback survey. Please, your responses are important for us to improve and to bring to you other webcasts going forward. For more information on this webcast and others, please contact Monica Alacon or myself, and other webcasts can be found on the STEMnet website. We have in the future 2 more webcasts this term. One on November 19th on AI and machine language, and December 10th on exemplars in biology. In addition, we have on November 5th another in our series of virtual research cafes 10.0. More information on both of these events will be forthcoming. And finally, Election Day will soon be upon us, and it's pivotal we exercise our fundamental human right to vote. In fact, over 45 million people, like myself, have already voted. November 3rd will likely be one of the most important days in the history of the United States, as it will determine our future and those who come after us. It is an understatement to say that the past nearly 4 years have been a challenging time for scientists, educators, teachers, and for all who pride themselves on speaking truths and who base their actions on facts. For today, we are at a crossroads where the backbone and central tenets of our democracy are constantly being challenged. Still, there is always hope. No matter how dismal things may appear, no matter how problem-prone we seem to be, no matter what reversals and setbacks we suffer, there is always hope. Hope is the thing that keeps us all going. In the CSU, not a day goes by we do not hope for something, that experiment to succeed, that paper to be accepted, that proposal to be funded. We bestow this hope upon our students and our colleagues. And while we do not always achieve what we set out to do, it is this hope that strengthens us and makes us, our students, our colleagues, our institutions of higher learning, a better and safe place. For hope does spring eternal in the CSU. So let us carry this hope to Election Day, for in the end we shall all overcome. Vote with your heart and vote your hearts out. Be safe and have a great day. I now turn over to Jennifer Wicks for a few closing remarks.
Thank you very much, Frank, and thank you for those reminders. I want to thank you for facilitating, moderating this session today, as always, and all of our incredible experts that were highlighted and showcased their work today. So thank each and every one of you for being here and your presentations. And we look forward to seeing each of you on another CSU STEMnet webcast very soon. As you see, we have the 2 coming up in November and December, and we look forward to seeing you then. Have a great rest of the afternoon. Thanks, everyone.